Good day, everyone. My partner and I worked on the topic effectiveness of UV LED at 265 and 275 nanometers for disinfecting laboratory personal equipment, face mask, and lab coats specifically against Staphylococcus aureus and Pseudomonas aeruginosa for bactericidal action. I would be introducing the introduction and materials and methods part of the project. The researchers decided to work around the topic UV LED as there has been much advancement throughout the years, and UVC has been used as water treatment for several decades now, but outside of this field, there has not been much advancement, specifically for surface disinfection, and it was discovered that specifically the UVC region with the wavelength of 254 to 265 nanometers has the highest inactivation rate for bacteria. Shortage of supplies and pollution brought about by the pandemic is the main reason why the researchers decided to work on this project. And the researchers aim to measure the effectivity of the UV LED disinfection on the chosen personal protective equipment at different time intervals, also to compare 265 nanometers against 275 nanometers. The researchers formed three hypotheses. The first one would be that the known concentration of bacteria will be approximately transferred to the personal protective equipment being tested. Second is that the 265 nanometers would have a higher bactericidal effect. And lastly, that the highest bacterial reduction would occur at the longest time interval, 180 seconds. The researchers chose the organisms Staphylococcus aureus as it is the most common skin infection, skin bacteria, and Pseudomonas aeruginosa as it was listed by the World Health Organization as a critical priority pathogen for research and development. For our materials and methods, the first step we did was to prepare our bacterial suspension with phosphate buffer. We did this according to the McFarland standard for both this for both the microorganisms. And after this, we spiked our personal protective equipments with both organisms and for the two different wavelengths. We disinfected said spiked personal protective equipment for 30 seconds, 60 seconds, 90 seconds, 120 seconds, and 180 seconds at a fixed current of 1400 milliamperes and 20 centimeter distance from the UV LED. After disinfecting them, we immediately suspended them in phosphate buffer, mixed them, and we did spread plating using 100 microliters of the phosphate buffer onto MSA plates for Staphylococcus aureus and blood agar plate for the Pseudomonas aeruginosa samples. After doing this, we incubated them at room temperature for 6 days, and after 6 days, we did colony count using the dark field colony counter. And my partner will now be introducing the results and discussion. Thank you. This is the prepared materials for the experiment that we did. We cut up the face masks and lab coats and set up the UV LED for 2 wavelengths.
Hello everyone, this is Anshul here. I and my group partner Nicole, we did the Capstone project. As she discussed the introduction of a project and methodology, I would like to start from the result part. So here in this slide you can see the picture of the plates, MSA plate and Cetramide plate for Pseudomonas aeruginosa and S. aureus. This was used as a positive control on the same day when we did our experiment. We streaked the pure culture on a separate plate to ensure that our bacterial culture was viable and the media prepared was good, so that if we get any deviations in our experimental results, we can find it out that there was no issue in the microorganism or media. So this slide is describing the table, which is presenting S aureus plated on MSA for 35 degrees Celsius for 48 hours after we disinfected the PPE, face mask and coat for the certain exposure of time. We can clearly see that for untreated plates for 0 seconds, we had maximum number of colonies. For the mask, it was 400 and it was 600 for the lab coat. And with the increasing time of exposure with UV LED at 265 nanometers, the number of colonies were decreased. And at 180 seconds, we can see that 100% reduction in colony count for the face mask, and we can see 95% for the lab coat. So we observed that number of colonies from the face mask were comparatively less as compared to the lab coat. The reason behind this could be the absorbance capacity of the lab coat is comparatively more as compared to the face mask material. So this was our analysis for this experiment. So here in this slide, we can see the same experimental setup results. The only difference here is 275 nanometer wavelength of UV LED, which was 265 in the last slide. So we can see there is a log reduction here for the laboratory PPE after disinfecting it for a certain amount of time, but comparatively it is less as compared to 265. We are having 86.25% decrease at 180 seconds, and for the face mask and for the coat it's almost the same, 86%. So from here we can conclude that 265 nanometer is more efficient for disinfecting PPE as compared to 275, and it was also one of the hypotheses for our experiment to compare two wavelengths for disinfecting S aureus and Pseudomonas aeruginosa. So here we have the picture at 265 nanometer plates, which when we did the experiment, we spreaded the PPE diluted in the buffer on the MSA plates for S aureus, and we can clearly see that while increase in the time of exposure, the number of colonies was continuously decreasing. At 0 second, the plate is quite yellow. It means that we have a lot of S aureus grown on the plate, so it turns out the indicator into yellow color. And while we go to 180, we saw that there was no colony at 182nd plate, and which is just clearly describing that there was 100% log reduction after 1 second of exposure at UV LED for 265 nanometers for S aureus. So here we have results for Pseudomonas aeruginosa. We repeated the same experiment for Pseudomonas aeruginosa and cetramide, but we were not getting expected results. We were again and again getting lawn instead of getting single colonies. So our professor suggested us, because of time constraints, to use directly the blood agar plates available in our laboratory. So we just plated blood agar plates with Pseudomonas aeruginosa live broth, and we checked the result after 48 hours and incubating that at 35 degrees Celsius, and we saw a decrease in number of colonies. And when we increase the time of exposure, and this table is clearly depicting our results, that there was 98% decrease in number of colonies for the face mask and for the lab coat as well. So from here we can conclude that our Pseudomonas aeruginosa was also disinfected at 265 nanometers when exposed to UV LED light. Here we have pictures of the results which we got for blood agar plate for Pseudomonas aeruginosa for 265 nanometers, and we can see that there is a decrease in number of colonies as we are increasing the time of exposure at 0 seconds. We got maximum number of colonies as compared to just 2 colonies at 120 seconds. Due to time constraints, we just did it for 265 nanometers, but we plan to do it for 265 and 275, same as for S aureus. But we were just able to complete it for 265 nanometers. So here I would like to raise few points which we made few changes from the experimental setup. We changed a different method of spiking bacteria. Instead of studying the absorbance capacity, we just spiked them all together in a phosphate buffer incubated with the live broth, and we got better results with the live broth as compared to the pre-diluted concentration of S. aureus or Pseudomonas aeruginosa. So we changed the method here and we got the expected results, better efficacy at 265 for S. aureus as compared to 275, and for aeruginosa we only got chance to study it at 265 nanometers, and we got decrease in number of colony count by increasing the time. We got just we The log reduction was to 98% after 120 seconds of exposure. So here we can conclude that this method of disinfecting PPEs is viable and can be used in future applications. And 265 nanometer would be better as it gave 10% higher disinfection as compared to 275. I would like to recommend that we can also increase the number of variables for this experiment if we have more time. Like we can change the distance from the UV LED, and we can also change the UV intensity. We already did it for 1400 mA. And one more thing, we just did it separately for two microorganisms. But if practically we talk about this experiment, so we have an infected PPE. We can have an infected PPE with different kinds of microorganisms. So I would like to recommend that we can make a consortium of viruses and fungal spores and bacteria, and we can spike PPE together, and then we can check the log reduction by using different methods of plating them, and it would give us better results and more practical applications. Thank you.